Hello, welcome to the last presentation for chapter 13. I have to apologize if I might pause every once in a while because I got allergies and sneezes going on today, so hopefully I can make it through. So this last lesson is going to be covering mutations and a tiny little blurb on a cool enzyme called reverse transcriptase. So before you dive too deep into the mutations, you want to make sure that you're relatively comfortable with the process of transcription and translation, because the mutations kind of apply that understanding to know the particular sequence of DNA gives you a particular sequence of mRNA, which gives you the particular sequence of amino acids. So if you're not quite comfortable with that central dogma yet, go back, spend a little bit more time on that, and then come back to finish up with this video. So when we take a look at mutations, like I said in the hemoglobin kind of introductory video, when we talk about mutations, mutations are changes in the DNA sequence. And those changes in the DNA sequence are transcribed into the RNA, which are then translated into the amino acid sequence. And that's the mutation, because the protein may or may not work properly. So this particular slide has three types of mutations, which are called base pair or point mutations, or base pair substitutions, where it's just one base change, kind of like what happens with the hemoglobin in sickle cell. So in this first panel here, we have what's called, or this is the normal, right? So here's the blue sequence is our double-stranded DNA, the kind of red sequence is our mRNA, and our proper amino acid sequence is methionine, threonine, arginine, asparagine, tryptophan, and aspartic acid. All right, so that's normal. But let's say a mutation happens. So it looks like this particular combination right there, we can see instead of a T, it got mutated into a C. Now this could have been an accident of the DNA polymerase. When it's doing the replication process, it just accidentally puts in the wrong base. And then when that DNA is replicated, that change is carried out on both sides. So if this is the template strand, because it looks like this is the template strand here, that C is transcribed into a G. So the codon, instead of saying AGA, it codes for AGG. That's a mutation. There was a change in the DNA sequence. And so AGA is converted to AGG. Now, if you go back and you look in your genetic code, which I don't have with me, but based on this picture, AGA codes for arginine. And if you look and find AGG, it also codes for arginine. Cool. So does this DNA mutation cause a change in the amino acid sequence? No, it doesn't. It's the same amino acid. So we call this a silent mutation. There's a mutation in the DNA, but the protein is unaffected. It's the same amino acid sequence. Because of that redundancy, remember, other than methionine and tryptophan, all the other amino acids have at least 2 codons that code for the same amino acid. Some of them had 6. So there's this ability to have a mistake in the DNA and the subsequent mRNA and still code for the correct amino acid. So that redundancy actually helps to prevent mutations and non-functioning phenotypes. These silent mutations also give genetic diversity. So if you are analyzing somebody's DNA, they may not have an actual disease or a phenotypic bad outcome, but they could have that change, and you can compare from person to person, which is kind of cool. All right, so let's take a look on that bottom left panel here. So we're looking at the same codon, but instead of the third base change, take a look, it's that middle one. So instead of AGA, we now get ACA. And if you look in your genetic code, ACA codes for threonine. Now, is that a problem? Well, yeah. Just like the sickle cell and the hemoglobin, it was a 1-letter change. Change, I think, was a glutamic acid to valine. And that made the whole, devastating disease of sickle cell anemia from one base letter change. If that amino acid is in a critical position of the folding and binding or active site of an enzyme, then you're going to have a big problem. This is a type of mutation that we call missense. So we have silent, where there's a base change but no amino acid change. And then we have a missense where there's a base change and an amino acid change. So it's a different amino acid gets put into place. And that can have varying impacts on the function of the protein. So if it's a similar type of amino acid, or if it's amino acid that's not in a very critical position of the three-dimensional folding, you could have a missense mutation and not really have any negative outcomes. But if it's critical, if it's in a critical folding position or part of the active site to catalyze chemical bonds, then you could have a severe phenotypic outcome. Okay, let's take a look at the last one in this bottom right-hand panel. So we're going to be looking at this combination. It's that middle base codes for, instead of UGG, which it was up in normal, it's now UAG down here in this mutated form. And take a look, you can go back to your genetic code if you have that open up with your book, and UAG is one of the stop codons. Yikes, okay, well that's not good. So here, instead of having our continuous complete protein, we have a truncated or cut short protein. And this is what we call nonsense. This is a nonsense mutation. So nonsense means the protein's not going to make any sense. It is cut short. Instead of having maybe 1,000 amino acids folding and making into a functional protein, maybe it has 50. A 50 amino acid sequence is not going to be anything functional. And if that's a very critical protein for cellular function, then maybe the cell might not even survive. Or you could have very, severe phenotypic outcomes. All right, so these are the three examples of base pair substitution, or sometimes called points mutation. So silent mutation, missense mutation, and nonsense mutation. The next slide is going to show you a couple more, but these are not point mutations, they're not substitutions. These are called frameshift. Now remember back when I showed you those 3 lines of reading frame, so if you shift the reading frame off by 1 or 2 bases, it completely changes the codon. Well, you can get that by either taking away or adding bases to your DNA. So again, that might happen with mutagens that are in your environment, or the sun rays that's mutating your DNA that can cause these frameshift mutations, or just a mistake in the DNA polymerase. So for this first one, I don't have the normal code, but let's say this whole TG is missing, and that's going to affect here's our start codon. We're missing the AC. It would normally be ACU, I believe, but then everything's shifted. So now we have a UAG, which goes for stop. So after methionine at stop, you're done. You get 1 amino acid and you're done. So this is a nonsense frameshift. Okay. The other one, they just took out 1 letter, but again, you're still changing your codon. So from that point on, every single amino acid downstream is going to be wrong, and it probably won't fold, and it'll be garbly-gook, and it's not going to make any sense. So this is also called a frameshift, but it doesn't have a super special name. It's just the wrong amino acid sequence. It's altered. So frameshift mutations typically are very detrimental in a cell's ability to function. With nonsense, you might get silent. You're not going to have a problem. With nonsense, you may or may not have a severe outcome. Even with nonsense, if the stop codon is just 1 amino acid early, you still might get a functional protein. But frameshift, usually not, because either you're going to get a stop somewhere in the middle, or you're going to get a complete different reading frame after that mutation. And here they're showing you deletions where parts of the DNA are taken out, but additions would be the same, could have the same outcome. So if you have an additional base, that's going to shove everything off by 1 or 2, and that's going to cause a frameshift mutation as well. All right, so those are our types of mutations. So we had our point mutations, which were silent, missense, and nonsense. And we had frameshift mutations, where you could have additions or deletions, shifting that reading frame, totally messing up the amino acid sequence. Okay. All right, the last slide that we're going to wrap up our chapter 13 stuff is a cool little enzyme called reverse transcriptase. So this is an enzyme that's not normally found in human cells. It was discovered in viruses that have an RNA genome. So any of you are taking microbiology as one of your courses for your prerequisites, you'll be learning all about this. So here's just a little bit of an introduction. So there are some viruses that when they enter your body, right? So here's our RNA virus. It's the red genome. They don't have DNA. They start with RNA. They come into your cell, and instead of being transcribed or translated into viral proteins, they're sneaky in that they have this enzyme called reverse transcriptase that takes their viral RNA and turns it into, or reverse transcribes it into DNA. So it goes from a viral RNA to viral DNA. Crazy, right? It just turns the whole central dogma on its head. And not only that, it then takes that double-stranded viral DNA that it just made, incorporates it into your chromosome, and that is forever a part of your chromosome. Now you have human DNA with a little slab of viral DNA in your chromosome, and it never comes out. It's a permanent part of your chromosome. So then let's say some time goes by and it just kind of hangs out in your chromosome, but then for whatever reason, it's going to follow the rules of our central dogma. It will get transcribed into viral mRNA. It will get translated into viral proteins, and it will start budding off new viral proteins because of the genetic code that's stored in your chromosomes. This is the way that HIV functions. Oops. These types of viruses are called retroviruses. So retro because they can do this reverse transcriptase, going from a viral RNA molecule to a viral DNA, incorporating the viral DNA into your chromosome, and then expressing that viral DNA as viral mRNA to make viral proteins, to make new viruses. It's pretty crazy. But it's so cool I had to tack it on because it kind of makes sense with this chapter. All right, so that wraps it up for chapter 13. So this week you guys were studying chapter 12 and 13, which kind of went together, DNA structure and replication with gene expression, transcription, and translation. Our last week, next week, we'll be covering chapter 6, which is cell communication. I will see you next time.